Hi, my name is Maggie. This tour of the Schaffer-Nishimura lab at Cornell was originally filmed for the community of catchers that have helped us speed up Alzheimer's research with our game, Stall Catchers. If you're one of those catchers, thank you so much for all you've contributed towards the fight against Alzheimer's. We couldn't do it without you. If you're new here, welcome. So many of us have been directly affected by Alzheimer's through a loved one, and those of us who haven't had those experiences have heard the heartbreaking stories. We would love if you would join our efforts. We are also able to have Mike Capraro join us on our lab tour. He's one of our super catchers, and he's made quite the impact in our community. We were so pleased with the opportunity to see the place where the magic happens, along with him. There is so much to see in this lab tour, so we've broken it up into 3 parts. This is the first installment, with the second and third soon to come. We hope you enjoy the tour of the Schaffer-Nishimura lab.
Hi.
Hi.
You're on Pietro's test.
Yeah, I'm the mule.
This is a little creepy.
Right?
I think we're probably doing the big one.
I don't doubt it. It's creepy for us too. So
I can imagine,
really
yeah.
Is this working? Can you see everyone?
Yes.
Yeah.
Yeah, okay.
I think we need a fisheye.
Oh, that would be good. Yeah, I bet there's some fancy filter for that. Okay, well, you can see where we are, and we're going to head down to meet Nancy. Thanks.
Okay, great.
All right.
Hi, Nancy. Nancy, have we ever met in person, actually? I can't. Yeah. Yeah, it's just been COVID. It's been a COVID long time. Yeah.
Hi.
Okay, so without further ado, if you want to take us in a direction, that would be great.
All right, cool. So we can first go into this lab. Biology. Some of us make viruses to label neurons in the brain. So this is a lab where all that happens.
Did you say make viruses?
Yeah.
Like, as in like the Wuhan lab?
No.
No.
Not quite. They're not infectious.
Not that kind of virus.
They're
Okay,
not infectious.
just clarifying. Okay.
Yeah, so basically all of that happens in here, more like molecular biology and
Yeah.
that kind of stuff.
So is this kind of like viral therapies where you use viral mechanisms to make adjustments in an organism that could help treat a disease, that kind of thing?
Not quite. Yeah.
Oh.
We only do it to label neurons.
Oh, label neurons. Okay,
Yeah, with
got it.
different colors.
I see.
But yeah.
But Pietro, you're correct. It's the viral vectors we use are called adeno-associated virus vectors. And those same viral vectors have been used in clinical trials led by Mike Capraro from Cornell Medical College. And in his case, they're used not to drive expression of a fluorescent protein to label neurons like Nancy was talking about, but rather they're injected into a particular part of the brain, and they drive expression of a protein that turns a group of excitatory neurons into inhibitory neurons. So neurons that normally, when they're active, they make downstream neurons more active, turns those into neurons that when they're active, they make downstream neurons more quiet. And it turns out that the part of the brain that this is done in is a part that projects to a part of the brain that's too active in Parkinson's disease. And so switching the kind of sign of the neurons in this one part of the brain has been shown to quell some of the symptoms of Parkinson's and again, work led by Mike Capraro from Cornell Medical College. So we make use of these same viral vectors. They're basically a way to be able to drive expression of a new protein or a new gene in neurons. And we use them to label cells, but they do have potential for therapies.
Oh, that's really exciting. Thanks for explaining that.
And they're called vectors because they're not real viruses. They can't actually reproduce and create more viruses. So basically, when you use them, they go into one cell and change the expression of proteins that you want in that cell, but they don't go on. So it doesn't actually make an infection in the same way as a disease-making virus would.
Got
All
it.
the
So it's okay,
replication machinery has been stripped out of the virus.
so it's okay to leave the door open is what you're saying.
Right.
Yes, yeah.
And yeah, you don't have to worry about having caught anything.
Right.
Right, okay.
That concludes the first installment of our Schaffer-Nishimura lab tour. The second one will be coming soon. In the meantime, we put some resources where you can learn more about stall catchers and the Human Computation Institute in the description below. Bye for now.